Okay, picture this. Imagine you are struggling to carry a heavy 19-liter container of water. That's roughly 42 pounds up several flights of stairs. Now, imagine having to do that 40 times a day. It sounds completely exhausting, right? Well, an average-sized tree pulls off this exact engineering feat every single day. It lifts massive amounts of water against gravity, and it does it entirely without a heart, without muscles, and literally without a single moving part. So how on earth is this actually possible? Welcome to this explainer. Today, we are going to unlock some of the best-kept secrets of the botanical world by zooming way, way in. We're going to transition from these massive, everyday botanical macrosystems down into the incredibly complex, in-depth molecular details. We're talking microscopic pumps, dynamic hydraulic valves, and lightning-fast electrical signals that make plant life possible. To make sense of it all, we'll be breaking this down into 6 logical steps. We'll start with the plant transport dilemma, look at cellular gates, dive into the physics of the xylem, check out the stomata, ride the phloem's superhighway, and wrap up with the dynamic Simplast network. Starting with step 1, the plant transport dilemma, and the challenge of living in 2 worlds. You see, vascular plants evolved to handle a really fundamental split. They exist in 2 entirely distinct environments at the exact same time. Above ground, their shoots have to harvest sunlight and carbon dioxide from the air, but below ground, their roots are busy mining water and minerals from the dark soil. Just to wrap our heads around the sheer scale of this transport problem, think about this number. A single maize plant transpires 60 liters of water in just 1 growing season. That's the equivalent of chugging 170 12-ounce bottles of water. And here's the kicker. If all that transpired water isn't perfectly replaced by water pulled up from the roots, the leaves wilt and the plant dies. It requires an insanely efficient delivery system. So to move all this water and nutrients around, plants essentially use 2 totally different, contrasting transport highways. First, you have the apoplast. This is basically everything outside the plasma membrane. We're talking dead cells, cell walls, and extracellular spaces. On the flip side, you have the symplast. This is the living, interconnected mass of the plant's cytosol, all linked together by these tiny microscopic channels called plasmodesmata. Which brings us to step 2, cellular gates, the molecular gatekeepers. Let's zoom right down to the cellular level to see how plants actually regulate what gets into their living tissues, especially when crossing the border from the dead apoplast into the living symplast. This is where we find a massive difference between plant and animal biology. Unlike our own animal cells, which rely heavily on pumping sodium, plants power their core transport processes by pumping hydrogen ions. They literally use ATP, which is the cell's main energy currency, to fuel proton pumps that physically shove hydrogen ions out of the cell. What this does is create a membrane potential. It basically acts just like a cellular battery. The plant then uses that stored potential energy to drive active transport or co-transport. For example, they'll use that hydrogen gradient to drag neutral solutes like sucrose or essential ions like nitrate straight into the cell against their natural concentration gradients. All right, step 3, xylem and transpirational pull, defying gravity through physics. We know how nutrients get into the cells now, but we still have to answer the big question. How does the water actually get all the way up the plant without a pump? Well, the absolute key to this is a concept called water potential, which scientists represent with the Greek letter psi. It's a physical property that combines solute concentration with physical pressure. And the golden rule you need to remember here is that free water always, and I mean always, moves from regions of higher water potential to regions of lower water potential. Kind of like how air rushes out of a high-pressure balloon into the lower-pressure room. And if you want proof that this movement is driven purely by physics and not some biological chemistry, we can actually look at the Q10 temperature coefficient for water uptake in radish seeds. Now, normally a Q10 coefficient around 2 or 3 means you're looking at a chemical or metabolic process, because biological reactions speed up when you add heat. But the calculated values for initial water uptake here are hovering right around 1. That proves the initial pull of water is almost entirely a physical mechanism driven by water potential. It is completely independent of the plant's metabolism. It's just pure passive physics in action. But don't let the word passive fool you, because this physical mechanism creates absolutely staggering forces. When water evaporates from the leaves into the dry air, it generates an immense negative pressure, or tension, inside the hollow, dead tubes of the xylem. We're talking a tension that can drop below -2 megapascals. To put that into perspective for you, the internal pressure of a healthy swollen plant cell is about twice the pressure of your car's tires. But the xylem is experiencing a negative tension so incredibly intense, it actually pulls the thick, microscopic walls of the xylem tubes inward. This perfectly lines up with the cohesion tension hypothesis. Water molecules are highly cohesive. Because of the slight magnetic attraction between the oxygen and hydrogen atoms of different molecules, they stick together like glue via hydrogen bonds. So, when that massive negative pressure hits the leaves, it pulls a continuous, unbroken microscopic chain of water all the way from the deep roots to the highest canopy. It literally gives the water inside the xylem the tensile strength of a steel wire of the exact same diameter. It is just mind-blowing. Moving right along to step 4, stomata, the balancing act. So this incredible upward pull is driven by evaporation. But a plant can't just evaporate water forever, right? It has to be controlled. Enter the stomata. These are microscopic pores on the leaves that act as dynamic hydraulic valves, constantly balancing the need to open up and grab carbon dioxide for photosynthesis against the very real danger of losing too much water and dying. The way these stomata open is an absolutely beautiful molecular cascade. First, blue light from the sun hits receptors that fire up those proton pumps we talked about earlier. The pumps shove hydrogen out of the guard cells that surround the pore. That sudden shift in cellular voltage causes potassium ion channels to fly open and potassium floods into the cell. Because the solute concentration just skyrocketed inside, the water potential drops like a rock, and water aggressively rushes in through specialized protein channels called aquaporins. The guard cells rapidly swell up with turgor pressure, bow outward, and dynamically snap the pore open. It is a masterclass in microscopic hydraulic engineering. Which brings us to step 5, phloem, the sugar superhighway. While water is moving up the plant passively, the sugars being manufactured up in the leaves have to be actively transported downward to feed the roots and the rest of the growing tissues. It's brilliant how elegantly the plant separates these two jobs. The xylem uses dead, hollow pipes to passively pull water upward using negative tension. But the phloem? The phloem moves sugars bidirectionally wherever they are needed, using living cells, and it's driven entirely by positive pressure. So where does that positive pressure come from? It relies on a chemoosmotic mechanism. The plant actively crams sucrose into the phloem using those hydrogen-sucrose cotransporters. As the sugar piles up, the water potential drops. Naturally, water from the neighboring xylem follows by osmosis, diffusing across the membrane to dilute that high sugar concentration. That sudden influx of water generates a massive positive pressure, literally pushing the sugary sap down the tube at speeds up to 1 meter per hour. And finally, step 6, the dynamic symplast network. Let's pull back for a second and look at the plant not just as a bunch of static plumbing, but as a living, breathing, deeply interconnected network that communicates across huge distances. This symplast network is constantly shifting. Those plasmodesma connecting the cells, we used to think they were just static holes, but it turns out they are highly dynamic. They open and close incredibly fast based on turgor pressure and chemical triggers like calcium ions acting as tiny molecular switches. On top of that, plants actually use nerve-like electrical signaling straight through the phloem to integrate the entire organism. They can alter gene transcription or respiration miles away from where a stimulus actually happened, at lightning speed. Fascinatingly, though, this superhighway does have a fatal vulnerability. Viruses actually exploit this exact system. They produce viral movement proteins that mimic the cell's own internal regulators. They hijack the plasmodesmata, force them to dilate, and then literally unfold their viral RNA to thread it right through these microscopic pores from one cell to the next. There's a great quote from the source material on this. It says, although they are often likened to the gap junctions between animal cells, plasmodesmata are unique in their ability to traffic proteins and RNA. And that really hits the nail on the head. The plant symplast is a total marvel of biology. It functions almost like a distributed nervous system that coordinates macromolecules, keeping the whole organism perfectly in sync. Which brings us to the end of our explainer today. But I want to leave you with one final provocative thought to ponder. If a plant can compute complex hydraulic pressures, actively manipulate membrane potentials with absolute precision, and transmit nerve-like electrical signals across hundreds of feet without a brain, what other profound biological intelligence is happening right outside our windows? Think about that the next time you walk past a tree. Thanks for diving deep into the molecular world with me today.